Hello everybody and welcome to today's video lecture. So today's video lecture is on direct methods of gene transfer. So in this video we are going to deal with the use of direct method of gene transfer and both physical and chemical methods of gene transfer. Okay. Physical and chemical methods. So let's move into the lecture. So first of all, what the gene transfer is. So it is the process of introducing a foreign genetic material into a target organism. So this may be target organism like a bacteria, or it can be cells. So we can collect cells and we can genetically modify it and then transfer the cells back into the organism. So this gene transfer can occur to cells or to organisms, usually bacteria, yeast, etc. Okay, so when we look into the types of gene transfer, they can be broadly classified into two, that is direct or vectorless gene transfer or indirect or vector mediated gene transfer. So in direct, it can further be classified into physical and chemical methods. So when we look into the physical methods, microinjection, biolistics, electroporation, laserporation, sonoporation, etc. are few major methods. And in chemical methods, calcium phosphate, DEAE Dextran, Lipofection, Cationic Polymer, PEG mediated transfer are few. So just for clarification, this lipofection and such kind of gene transfer methods uses a carrier molecule, maybe a lipid or a polymer, as in cationic polymer. So when a carrier molecule is used to transfer the genetic material, it can sometimes be classified classified or seen as a vector, because vector is something that carries the genetic material. So here the lipid molecule may carry the genetic material, right? So here it can be seen as a vector, but commonly it is classified into chemical method of gene transfer. And in indirect or vector mediated gene transfer, it comes plasmid mediated, viral mediated, and so on. Live vectors would be used. So this is the general classification, although only few are mentioned here. We will discuss even more. So let's move into the next slide. So here we have some important terms. I just want to clarify these terms so that for no confusion exists. So transfection. So transfection is the method that we are going to use, like the process of introducing nucleic acids such as DNA or RNA into the cells using non-viral method. So this physical and chemical method, it actually comes under the transfection. And this transformation generally refers to when genetic material is incorporated into a bacteria. You just take an example, bacteria. So this bacteria get transformed, it gets genetically modified. So this genetically modifying of bacteria, that is called the transformation. Okay, and then transduction. When we come to transduction, this virus, maybe a bacteriophage or any such virus, aids in the transfer of genetic material from from the virus into the bacteria. So this process is called transduction. And in conjugation, the bacteria transfer occurs between the bacteria, right? So F pili or conjugation pili will be established between two bacteria and the genetic material will be transferred. So in conjugation, this type of conjugation is actually called as the horizontal conjugation. So in horizontal conjugation, the transfer of genetic material will occur in the same generation. So in the same generation, the transfer of genetic material is occurring. But whereas in vertical gene transfer, the daughter cells get the genetic material. Okay, the genetic material will be incorporated, that is being incorporated into the parent cell, get divided into the daughter cells, and daughter cells get the genetic material. So this is the vertical transfer, as this name suggests. This is vertical way, this is the horizontal way, right? So this is the horizontal gene transfer. So these are few terms that you should be thorough with to understand the topic in a better way. Now when we come to transfection methods, it can be of two types, majorly of two types, that is transient transfection and stable transfection. So let's look into the transient transfection, that the DNA is not integrated into the genome. So maybe if this is a bacterial cell and this is the genome of the bacteria, you can maybe introduction of a plasmid like this, you can see it like. So this plasmid need not get integrated into the genome, right? So this kind of introduction is called transient. That is, the genetic material whatsoever we are introducing is not getting incorporated into the genome of our target organism. And since it is not getting integrated into the genome, it will not be transferred or passed on to the progeny, that is the daughter cells will not have. So and this method also do not require perfect selection methods, like we are introducing it into the cells, we just need to make sure that the cell acquired the genetic material. We need not check whether it is getting integrated into the genome or not, it doesn't matter. Then both DNA vectors and RNA can be used for transient transfection. So DNA can be incorporated or RNA can be incorporated, anything can be incorporated. And high copy number of transfected genetic material results in high level of protein expression. And in stable transfection, the transfected DNA integrates into the genome, that is the DNA is getting integrated into the genome. Maybe this is the DNA and it is getting integrated into the genome. So this integration ensures that the progeny daughter cells also receive the gene of interest or target gene. Okay, it is carried from one generation to the other. And it requires selective screening to ensure that the target gene is being incorporated into the genome of the organism. And since the organisms that we usually use have the DNA as its genetic material, only DNA vectors can be used for this stable transfection. And single or low copy number of stably integrated DNA results in lower level of protein expression. So hope you understood this, the transient transfection and stable transfection. Now let's look into the factors influencing transfection. So one factor that influences transfection is the cell type. Maybe you take a plant cell and an animal cell. So this plant cell is having something called the cell wall around it. So the transfer of a genetic material from outside to inside, it requires to penetrate the cell wall layer. So cell wall layer need to be penetrated, and in case of animal cells it only have a plasma membrane covering. So the transfer of genetic material will be comparatively easier when we compare it with the plant cells, right? It need not penetrate the cell wall, the thick cell wall, and reach the interior of the cell. Then plant cell health. So here the health of the cell is very determinant, because when we use different transfection methods, maybe chemicals or physical methods, if the cell it is damaged, it is very weak, then when we use a transfection method, the cells may get destroyed and then we will not get the required outcome. But if the cells are healthy, it can protect itself from the external stresses that will be caused due to transfection. See, the cells are natural things. So when we do some manipulation or genetic manipulation, we are giving stress to the cells. So cells must be healthy enough to endure the stress. Then the confluency. Confluency is the density in which the cells are present. Okay, so where we have two Petri plates or culture plates with one with very less confluence. This is very less confluency of cells are present because very less number of cells are there. But in the second Petri plate we can see even more number of cells, right? So this Petri plate is said to have high confluency of cells. Okay, and this Petri plate is said to have lower confluency of cells. So here if we are inducing some genetic changes, we are doing some transfection methods, what happens? It is this transfection method doesn't ensure that 100% of the cells get transfected or genetically modified. No, that do not happen. So the chances of getting the cells getting transfected decreases when the confluency is low, right? And the chances of getting cells transfected when the confluency is high also increases because there are many cells and we are doing this procedure on this culture plate, and there are more chances that more cells get transfected. Now when we look into the serum is something in which our cells are grown, right? So this serum contains many components, and one of the components are ions like calcium, and many ions are there. So when we use the chemical methods or when we use the physical methods like electroporation and so on, so both this type of method should be taken by considering the type of serum. So what happens, I will just explain. If we are taking a chemical method and when we use the chemicals, what if the pH of the serum changes? Then what happens? The cells will die, right? So this should be considered. And if you are taking a physical method, so let's say electroporation. So in electroporation we apply the electric field. So when we apply the electric field, if the serum contains ions that separate when electric field is applied, so there is a positive and negative electrode that will be there, and this components of the serum is getting separated. The positive ones will move to the negative electrodes and vice versa. Then what happens? Then the serum stability is lost, right? When the serum stability is lost, the cells tend to die. So this should be avoided. So whatever transfection method we use, we have to make sure that the serum component is not getting affected. Now time is required for the cells to reach the confluency. To reach the confluency, time is required, 1. Then 2, the time or duration for which the transfection process is applied, that also depends upon the time. See, if we are doing some electroporation, the time of applying electric field, it is very determinant. So if the time is more or if the time is less, we will not get optimum transfection, right? And then DNA quantity and quality. In DNA quantity, if the quantity of DNA is very low, can we ensure that all the cells will receive the DNA? No. So it's logical. And the DNA quality, if the DNA have more of A, T base pairs. So what happens? If GC is present, they have 3 hydrogen bonds between them, right? But compared to this, if AT is more, it have less number of hydrogen bonds. Only 2 hydrogen bonds are there. So what happens? When AT is more, the stability of DNA is decreasing, right? So what happens? Since the stability is decreasing, the quality of DNA is being compromised. So these are few factors that influence the transfection. Hope you understood. Now let's see where our transfection occurs and where we apply the transfection and all. Okay, so here we have a gene of interest, and here we have the plasmid of the bacteria. So here we integrated the gene of interest into the plasmid. Okay, successfully integrated. We integrated using DNA ligase. First we used restriction enzymes to break the strands, and then we used DNA ligase to ligate the gene of interest into the strand plasmid. Now we need to transfer this plasmid into our bacterial cell, right? This is called transfection. Here we use the methods of transfection. Here it can be physical or chemical methods, okay? So this is where the transfection occurs, and we are going to study into detail what. So here, first of all, we have the physical methods of transfection. Physical transfection methods. So let's take microinjection first. So microinjection is one of the physical methods that is used to transfer gene or the genetic material using glass needle or a micropipette. So here which we see the instrument, this is called the microinjection, and here this is the glass needle, or we use glass micropipette. So this was introduced by our Marshall A. Barber, and he introduced it in this 19th century. So he is Marshall A. Barber. So principle of microinjection, it's like, you directly inject the genetic material into protoplastic cells. So as you know, we are directly injecting the genetic material, say, into the cell. If you just think, you may understand that the plant cells which have the cell wall is not that suitable in this method, right? So this is what I meant when I said you have to select the required transfection method that you need to use, and it depends on the type of cells and serum, etc., etc. Okay, so here we have this microinjection, that is our glass needle, which is about 0.5 to 1 micrometer in diameter, which will be injected into the cytoplasm of our cell. So it is used to incorporate or inject DNA into large cells, because only large cells can be held by a holding pipette. A holding pipette is used to hold the cell, and then from the opposite side we inject the cells with the genetic material. And this setup, that is this micro injection plus the optical microscope, this setup is together called the micro manipulator. And this computerized control of holding pipette, needle, etc. has increased the efficiency. Nowadays there are highly sophisticated machines for doing microinjections. So let's look into the methodology. So here I have given the notes of the methodology. You can read through the notes. I will just go through go to the diagram and then explain it. So here we have the cells. So this is the cell and this is the holder. So this holder uses a suction force to hold the cells in place. So this cell is held in place. Then after it is being held in place, a micro injector pipette, a glass pipette, a micro injector needle will be introduced into the cells very gently, and then the hydro pressure will be given, and the pressure will be given. So due to this pressure, what happens? The genetic material get transferred into the cells. Okay, so the genetic material get transferred. Once the genetic material will get transferred, you can see the morphology of cells. See, first the cells were like this, but after the genetic material get transferred, the cells become a little swollen, right? So you can see that. And then after injection, the needle is pulled out very softly without breaking the cells. And then the cells are left out for recovery. So this is the gene transfer method by microinjection. It is very simple to understand. So when we go to IVF, usually this kind of methods are being used. We will have a mother cell, mother egg cell, and into the egg cell the sperm nucleus is being injected. So here the microinjection, it can be done into the cytoplasm or it can be done into the nuclear region depending on your need. Now advantages, the frequency of stable integration of DNA is far better as compared to other methods, and the method is effective in transforming primary cells as well as cells in established cultures. The DNA injected in this process is subjected to less extensive modification. There are no much modification. We are taking the genetic material and injecting it. We are not modifying it in any way. So no modification occurs. Then more precise integration of recombinant gene in limited copy number can be obtained. And when we look into the disadvantages, it is costly because we are using equipments and all, so it's costly. It is skilled people are required. See, the person who is doing this selects the cells. So what if the cell that he selected is not good, not healthy, then the result will not be good. So it is his decision that matters a lot. A lot of skill is required to identify the healthy cell through the microscope and take the cells. And it is more useful for animals. As I said, the plant cell is having cell wall and it's difficult to penetrate the cell wall using the syringe. The needle tip may break off. Then embryonic cells are preferred for manipulation, as I said in IVF. The knowledge of mating time, move site, recovery, everything is essential. We have to know the cell cycle completely so as to know when to inject the genetic material, right? So that is important. And methods is useful for protoplasts and not for wall cells. Even so, it is not used for wall cells. Some plants have been successfully transfected using these methods. For example, tobacco, alfalfa. Then process is applicable, can be applied to plant cells as well, but animal cells are more preferred and more common in use. And transgenic animals can be quickly made, and this procedure is important for gene transfer into embryonic cells. Applied to inject DNA into plant nuclei, it can be done, but it's not favored, okay. Then microinjection is potentially a useful method for simultaneous introduction of multiple bioactive compounds such as antibody, peptide, RNA, etc. We can take the suspension or mixture of all these things and we can introduce multiple bioactive compounds in a single injection, and that can be done into the target cell. Now let's move into the next, that is biolistics, gene gun, particle acceleration, microprojectile bombardment. There are many names for this method. So this is also one of the physical methods, and the gene it is used to transfer the gene. Okay, we are studying that, to introduce the foreign genetic material into cells or tissue using high velocity particles. So here what we use is like a gun. It is like a gun, and it's like a gun, and we use high velocity particles. We shoot it into the cells, and this high velocity particles get penetrated into the cells, just like a gun. So here it was originally done on, developed on a Crossman air pistol, and this pistol was initially used to fire tungsten particles. So this tungsten will be coated with the genetic material, right? And this tungsten particles will be fired into the cells. So the cells will acquire the genetic material. So this is how it was done initially. And it was invented by John C. Stanford and E.D. Wolf. So he is Stanford and he is Wolf at the Cornell University, and Ted between 1983 and 1986. So initially it was developed for sufficiently large plant cells, that is onion cell transformation was the experiment that they did. So it was developed for that. So as you can see, the onion cell, it's a plant cell, and we are shooting, it's a gun, it is having some power, right? So it can penetrate the cell wall, that double hard cell wall of the plant cell. So it can be used for plant cell. That's what I wanted to say. So here principle, the genetic material is directly in short, you can say it is short, right? It is a gun, so it is, we are shooting the genetic material into the target cell with microcarrier particles. The microcarrier, it acts as bullet, microcarrier plus the genetic material, so that is the bullet. So the microcarrier is usually gold or tungsten, and it is coated with the genetic material. They are inserted into the target cell using high velocity stream generated by either electric discharge or helium pulse. Usually nowadays our helium is being used. So helium pulse will be given, and that is the power or energy with which the gun is being shown. So high velocity stream shoots are done. Okay, so then once inside the cells, the genetic material will separate from the microcarrier, and it will be taken up by the machinery of our target cell. So when we look into the instrumentation, initially the gunpowder was used in the place of helium. We just discussed the helium or electric pulse will be given, right? But initially when it was developed, gunpowder was used to propel the microcarriers. So this gunpowder system was later on replaced by high pressure helium systems for particle acceleration. So this using helium in place of gunpowder have improved the transformation efficiency to a greater point. So we know helium is a noble gas, and unlike gunpowder, it is gunpowder is chemical, right? So this change has increased the efficiency. The achievements in technology have led to development of improved gene guns like electrostatic, pneumatic, and compressed gas gene guns. All these variants are developed, and PDS 1000 helium, I will show this machine, or was it here? Yeah, this one. This is the PDS 1000 and Helios gene gun, the first one which we saw. I will show, the picture is coming again, so I will show it. Then here the Helios gene gun, it is a handheld device. Okay, and this PDS, it also uses the pressurized helium gas to accelerate the microscopic gold or tungsten particle that is coated with the genetic material. So let's move into the methodology. See, from here you can take the notes. I will just explain with the diagram. So this is the setup, and as I said, this is the PDS 1000 and this is the handheld Helios gene gun. Okay, so since it is handheld, we can use it for in vivo gene transfers, and you can just note that too. So this is the setup. Here, this is the tube where the helium gas helps in the acceleration of the particle. This is the ruptured disc, and this disc breaks open or opens up to allow the pressure to fall into this thing. This is the microcarrier. This is the microcarrier. The small ones are microcarriers, that is gold or tungsten plus genetic material. And this blue one, that is the macrocarrier. So the microcarriers, we can call it microprojectiles, so you just don't get confused. So the microprojectiles will be loaded in the macrocarrier, and when the pressure falls on this, what happens is that the macrocarrier falls into the stopping screen and it stops there. But due to the pressure, the microprojectiles leave from the macrocarrier and it falls with that pressure. With the help of that pressure, it falls on the cells. So due to that pressure, the cells, the microprojectiles are shot into the cells. So the cells get transformed. The genetic material will be incorporated into the cell, and the transfection occurs. So this is how the microprojectile bombardment occurs. So here we have another, a bomb-like thing for the same, but the preparation is a little different. So we will look into it. Light-triggered nanoscale biolistics. So what happens here is that a nanobomb synthesis is done. So here the vapor bubble VB plus the nanoprojectile, it will be treated with laser. So a nanobomb will be formed. When that it is treated with the laser, its temperature increases. It increases to such a point that at any time it may get released. So that is this condition, and in this condition the nanobomb will be fired into the cells. So it will penetrate the cell plasma membrane and it will enter the cell. And once it is inside the cells, it reaches a threshold and the release of nanoprojectiles occur. So this is another method, and this nanoprojectiles, it get incorporated into the genetic material and expression of the gene occurs. So this is called light-triggered nanoscale biolistics. Advantages: requirement of protoplast can be avoided. As I said, plant cells can be directly used because enough pressure is there for the genetic material to penetrate the cells. So no problem occurs. Walled intact cells can be penetrated, and manipulation of genome of subcellular organelles can be achieved. This technology has even allowed for modification of specific tissues in situ. That is, I told you, the Helios gene gun, it can be used for in situ transfers, transfection method, and it have very high efficiency and it is much similar. It is like just shooting a gun. Okay. Then the disadvantage comes like it is random and it requires equipments, as we saw in Helios gene gun or 1000. The equipment is much required. Then shallow penetration of particles may occur. Then cell damage can also occur, and the DNA must also be able to the tissue to incorporate the DNA must be able to regenerate, because when we shoot, many holes will be created on the cell surface, right? So this cell surface holes must be able to regenerate and heal itself. Then the equipment itself is a little expensive. So these are the disadvantages. And when we look into the applications in plants, we have seen, this technique has been used successfully to transform soybean, cotton, spruce, sugarcane, papaya, sunflower, etc. The particle gene gun has also been used with pollen, early stage of embryoids, meristems, etc. in situ type of transfection, and it can be also used in place of agrobacterian tumefacients, right? Because it can be used in situ mode. So instead of infecting a plant with agrobacterian tumefacients, we can directly transfer the gene of interest using the gene gun. And in animals, the gene guns also have been used to deliver DNA vaccines, then plasmids into rat neurons, then labeling subsets of cells, and transforming of elegans and all such on. Now let's move into next, that is electroporation. It is also called electropermeabilization. So electroporation is another physical method for introducing the genetic material, protein, drugs or any other desired molecule into the cells for various purposes with the application of electric pulse. So here we use the electric pulse to create temporary pores. So why these are called temporary pores means, like this pores that is being generated due to electroporation will heal in itself. So these are temporary pores. Okay, so Newman, he is the guy who, he is the sir who developed the concept of electroporation in 1982. So let's move into the principles. The application of electric pulse into the cell cause disruption of the cell membrane, and the pulse changes the electrical potential. So this disruption of cell membrane occurs due to the change in electrical potential across the membrane, leading to temporary pores in the phospholipid bilayer that forms the cell membrane. These pores allow molecules to pass, and when the electricity is turned off, the pore starts to close, that is the heal. Okay, and the molecules will be trapped inside the cells. So it can be reversible electroporation if the defect heals, that is the healing process, if it occurs, it is called reversible electroporation, and if the healing do not occur, it is called irreversible electroporation. And if the healing do not occur, that is the cells with pores, what happens? The cell will die, because the contents inside the cell get outside and water get inside, so the osmolarity is disturbed and the cell death occurs. Okay, so this is the thing. And as we know, the plasma membrane, right, the plasma membrane is made up of lipid bilayer. So we have lipid bilayers. So this lipid bilayer, it is having charges, right? The hydrophilic head, hydrophobic tail, we have seen all those things. So this property of the membrane is actually what causes the pore formation. Okay. Now let's move into the instrumentation. These are few instruments that are generally being used. The Gemini twin wave electroporation machine, then intramuscular electroporation device. This is some kind of in vivo technique, we can say. Then variables involved. So in general, the field strength, that is, you can see the field strength is the voltage. Field strength means the voltage that is required, to induce the membrane break. So the membrane, some charge separation must occur, right, for this membrane to break down or a pore to form. So for this charge separation to occur, a specific voltage must be applied, right? So this is the cell. Okay, so this specific voltage is called the field strength, and the voltage must be optimized before starting an experiment, because if the voltage is not optimized, then it may lead to irreversible electroporation and cell death may occur. Then pulse length is the duration of time the sample is exposed to the electric field. That is the time for which the sample should be exposed to that particular field strength for this potential difference or pore formation to occur. That time, that is pulse length. So if the time is more, if the time is less, then also the proper pore formation may not occur or the cell may die. So both these variables must be optimized before starting the experiment. And methodology, these are the notes. You can go through the notes. I will go to the diagram to explain it. Okay. So here what happens is that the cell membrane is there, right? So we have charged molecules. This is our cell. So the genes to be introduced is in the media that we are taking. And the electric field, here it is mentioned that 3000 to 4000 volt per centimeter for less than 1 minute. So such electric field will be applied. So within that time, this charge segregation will occur, and due to this charge segregation, pore formation will occur. When the pores are formed, the genetic material or gene of interest that is there in the outside of the media cell, that is in the media, it get transferred into the cell, transferred inside the cell, and it get trapped. And when the electric field is removed, then the pores that was formed will seal, and the genetic material is trapped inside the cell. So our aim is achieved. So that's what happens. And here when we look the migration of ion, all these things we said, but yeah, I have a point here, that is pre-pore. Once the critical field is the field in which the rapid localization of ions occur, and this separation of ions, right here, this is the critical field. The critical field is the electric field voltage at which this separation and pore formation occurs. So during that there is rapid rearrangement in the lipid, right? That is why since the rearrangement in the lipid is occurring, that is the reason why the pore forms, and this resulting structure, that is called a pre-pore. So this structure where the pore is occurring, that is called the pre-pore structure. So that is the methodology. So this is the methodology. So it is the same thing explained in a diagram. No, it's mentioned. Okay, so let's move to the factors influencing electroporation. The success of electroporation depends greatly on the purity of plasmid solution. The plasmid solution must be pure. What if ions are there? Then these ions will participate in this electrical field movement, and the ions also, the salt, all these things will move inside, and it will cause a lot of confusion. So the purity of plasmid solution is very important. Then the solution with high salt concentration might cause electrical discharge. See, these are the cells, and in that we have the solution that we are taking is having many ions. So what happens is that in place of the ionic separation that should occur in the cell surface, when we apply a potential, the ions inside the inside the solution, these ions will participate in this separation, and these ions will get separated rather than the ions in the cell. So this problem can occur. Then cells that are arrested at metaphase stage are more suitable for electroporation method. Why? Because in the metaphase stage what we have is the cell without any nuclear envelope. There is no nuclear envelope. There are genetic material that is arranged in the metaphase plate, right? So there are cells, and then the cells they contain the genetic material. So the cell gene of interest need not penetrate the nuclear membrane. So hence, this cells that are arrested in metaphase stage is the best suited for electroporation. Then protoplasts are used for electroporation for plant cells. As we have already discussed, the plant cell walls are thick, so it restricts the movement of DNA. Then electroporation was originally developed for protoplasts, so it is not suitable for plant cells because it has cell wall. That's what we are saying. So if we are taking plant cell, we can remove the plant cell wall, isolate the protoplast, and then apply the electroporation. It's okay, whatever method, all the methods that we have discussed before. If we are using plant cell, we can isolate the protoplast from the plant cell and do the methods, and it is perfectly fine. Can you do it that way? Then transgenic mice, it can be produced. Now let's look into the advantages. These are very simple, convenient, rapid, and it's also cost effective. It is non-invasive, non-chemical method, and it does not alter target cell. Since chemicals are not used, the mutations or anything, such kind of things will not occur. Then transform cells are at the same physiological state. The fast and easy method, it is not toxic, efficiency is very high. Because if we optimize the electric field, the efficiency will be optimum. The electroporation technique can be applied to much wider selection of cells. Many cell types can be used. Then the disadvantages are, under normal conditions, the amount of DNA delivered into the plant cells is very low. Efficiency of electroporation is highly variable depending on the plant material and treatment condition. Generation of plant is not very easy, and particularly when the protoplasts are used. Okay. Now, when we move to the applications, only protoplasts were used for gene transfer by electroporation. We discussed that. So, if we are using plant cells, we can use it by proper pre and post electroporation treatments. Then transgenic plants like cereal, rice, maize, wheat, etc. were developed. Then electroporation is highly efficient for introducing foreign genes into tissue culture, transformation of bacteria, yeast, etc. Chemotherapy, gene therapy, diagnostics, etc. For all these we can use electroporation. Now let's move into the next, that is our magnetofection. So in magnetofection, we use magnetic force to deliver the genetic material. Magnetic force is used to deliver the target gene of interest into the cell. So here the nucleic acids are first associated with magnetic nanoparticles. So there we have this round thing, the magnetic nanoparticles, and we will attach the nucleic acid to the magnetic nanoparticle. Then we will keep the cells on a surface, and below that we will give the magnetic field, and the cells, the magnetic nanoparticle will be accelerated into the cells due to the magnetic field that is applied beneath it, right? So let's move into the working. So as I said, the working is what I just said before. So the cells, it will be grown, adherent cells are used. Okay, adherent cells are commonly used, not the suspension ones. So it's common sense, right? You can understand, when the suspension cells are used, the cells will be freely floating. So what happens, targeted acceleration of the magnetic nanoparticle into the cell become difficult when we apply the magnetic field. So that is the reason why we use the adherent cells. So first of all, we will take the magnetic nanoparticle, and we will oscillate the magnetic field or magnet. And what this oscillation does is that it will move the magnetic nanoparticle on the surface of the cell, and this movement triggers the endosome formation and endocytosis of the magnetic nanoparticle occur. Once endocytosis occurs, proton sponge effect takes place, that is, the H plus ions get incorporated. First of all, the Cl minus ion get incorporated, then the water get incorporated, and due to the pressure created by both these ions, the endocytic vesicles, it will break, it will rupture, and the genetic material with the magnetic nanoparticle will be released. The genetic material will go into the nucleus or wherever its target is, and the magnetic nanoparticle it get degraded, and the genetic material that is transferred, it get transferred into the nucleus, it get maybe incorporated into the genome, then it get transcription occurs, then it get translated, and a protein will be formed. So all these things can occur. So this is the way by which the working of magnetofection is. Okay. Now the advantage is that it is very rapid and it is very efficient. Then high transfection rates can be obtained performed in presence of serum. If the cells, if the adherent cells, it is present inside the serum, and even if the serums are containing any ions or whatever, it doesn't really matter, because the magnetic field is being given and the nanoparticles will be targeted into this and into the cells, and the endocytosis of the magnetic particle will occur. So it doesn't matter. Then the disadvantage is that it requires adherent cells. Suspension cell cultures need to be immobilized or centrifuged, and it should be made to adhere into the surface of the cells, surface of the culture plate or whatever, for doing this method. Now let's move to the heat shock method. So here the mechanism is the rapid temperature change will create temporary pores on the cell membrane, facilitating the entry of genetic material into the cell. So important considerations are temperature and cell viability. See, if the temperature is too high or too low, it may affect the cell. And the cell viability, whatever method we are applying, we have to make sure that the cells are viable. The cells are not getting damaged during the process of transfection, while we apply the different transfection strategies. So we have to select the transfection strategy that that facilitates the most viable condition of our cells. So the procedure here, we have to first prepare the cell, make the cells competent, and this is done by calcium chloride. The cells are treated with chemicals like calcium chloride to increase its competence, that is, it becomes more ready to absorb the DNA. Now the cells are mixed with DNA. So since the cells are made competent, then the prepared DNA is mixed with the component cells and incubated on ice to allow for initial binding. So the calcium chloride treated cells will be mixed with the genetic material and it will be incubated on ice for a very brief time. So the initial binding, that is the genetic material, comes in contact with the cells. Okay, that's what happens. And then the cell is rapidly transferred into a water bath at 42 degrees Celsius for a very short time, that is 30 to 60 seconds. And then after that, it is rapidly transferred quickly transferred on the ice to stabilize the membrane. If it is kept on the temperature, if the cells are kept in 42 degrees Celsius for a longer period of time, the cells will get damaged, it will lose its viability. So what we do is that we keep it on the ice for the cells to recover, for the membrane to recover, or for the membrane to stabilize before we do further steps. So this can be repeated for 2 to 3 times to ensure that complete intake or maximum intake of the gene of interest occurred. So this is the method. Now we move on to laserfection or opto-injection. Here the procedure uses laser light to transiently permeabilize a large number of cells. So this method, the major advantage is that we can use a very high number of cells, and number of cells used that can be transfected at a time is very high, and various substances including ions, small molecules, or maybe RNA, protein, etc. can be octo-injected, that can be injected into the cells. And here what happens is that we take the intact cells and we take the impermeated nanoparticles, the nanoparticles with our genetic material or target molecule, and then we give 532 nanometer laser beam. So what happens when we give the laser beam, it causes pore formation or transient permeabilization of the membrane surface. So here the membrane pores or that permeability is created, due to which the nanoparticles get incorporated into the cell, and when the laser is removed, the cells heal and the material get incorporated. So we get the target molecules inside the cells. This is what happens. The advantages of this method are transfection efficiency is much higher, and ability to make pores at any location of the cell, then work with many cell types, fewer cell manipulation is needed. Disadvantage is that it is very expensive because we use laser microscope systems and all, and the cells need to be attached to the substrate for doing the method. So our next method is cell squeezing, and this method is a kind of mechanical method, and it was introduced in 2012 by Armand and his team. So what happens here is that the cell is taken in a suspension of our compound of interest or gene of interest or whatever. So the cell is then passed into a very small orifice such that the cells have to squeeze through it. So this squeezing causes the cell membrane to produce mechanical damage, that is pore formation or mechanical damage to the cell membrane occurs, and through this damage our gene of interest or the compound of interest enters the cells, and then the cells will acquire this gene of interest or whatever, then the cells will heal and transformed or transfection is successfully done using the cell squeezing method. So this is the cell squeezing method. So here there no toxicity or off-target effects occur. As we can see, we are not using any other chemicals or any other electricity or any other physical methods like magnet or nothing. We are not using anything. So here toxicity, it eliminates the possibility of toxicity. Then off-target effects, we are making the cells one by one go through this narrow orifice, making sure that the cells are getting transfected, so no off-target effects occur. And this is as I mentioned, it is mechanically deformed. The cells will be mechanically deformed, and through this deformed membrane only the target material get incorporated. Then transient holes will be allowed the diffusion of material. So anything can be transfected, maybe a protein or RNA or genetic material, anything can be done. So this is a mechanical method of transfection. So this is the same thing. Now let's move into the chemical method of gene transfer. So chemical method, these are the major chemical methods: cationic liposomes, calcium phosphate, cationic polymer. So we will see different types of chemical methods. We will see even more. Only 3 are mentioned here. So first let's look into the calcium phosphate method. It was the first chemical method used for transfection, and it was developed by Graham in 1973 for introduction of adenovirus DNA into the rat cells. So the principle involves the calcium phosphate facilitate the binding of the condensed DNA in a co-precipitate into the cell surface. So what happens here is that there is cell and the DNA, the genetic material, will co-precipitate on the cell surface along with the calcium phosphate. So when this calcium phosphate plus genetic material precipitates on the surface, the endocytosis of the calcium phosphate plus genetic material occurs and it gets internalized into the cell. So this is what happens. So when we do this precipitation, while making the precipitate, we provide adequate aeration. So what happens when we provide good aeration? The precipitate formed will be much minor, like fine precipitates will be formed. So the smaller the size of the precipitate, the easier it will be for the cells to incorporate it inside the cell, right? So that's why proper aeration are done. Now methodology, it's the same that I said. The calcium phosphate plus DNA, it will be made into as fine as possible, and it will be endocytosis will occur, and the cells will intake the genetic material or gene of interest. And then what happens? The proton sponge effect that we discussed earlier, the proton sponge effect occurs, the endocytic vesicle ruptures, and the genetic material will be released. So this is what happens. So it is very easily available, simplest, and many cell types can be used, and it can be used for both transient and stable transfection. Disadvantages is due to the variability and due to sensitivity to slight changes in pH, temperature, buffer, salt concentration, etc. Then potential issues with reproducibility. The way in which one cell get transfected, we can't exactly reproduce the result in another cell. So that is a problem. Then cytotoxic to certain cells. Then it is unsuitable for in vivo transfer, cannot be done. Okay, most of the chemical method it cannot be done with in vivo. Then it cannot be used with RPMI media. RPMI media means Roswell Park Memorial Institute media. It cannot be used with that. Then relatively poor transfection efficiency compared to other chemical transfection methods. Now let's move into lipofection. So this is a lipid mediated transfection technique used to inject genetic material into the cells. So here we use liposomes. So liposomes are microscopic vesicles. They are vesicular type with a 20 nanometer of diameter, or it may extend to few micrometers. The diameter may be up to few micrometers that can easily merge. So since these are lipids, and our plasma membrane is also a lipid bilayer, right? Lipid bilayer. So it can easily get incorporated into the cell membrane. So depending on the size of liposome, it can be categorized into following classes. There are mainly 6 classes of liposomes, that is small unilaminar vesicle SUV with a size of 20 to 100 nanometer, large unilaminar vesicles LUV with more than 100 nanometer size, giant unilaminar vesicle with more than 1000 nanometer, oligolaminar vesicle with a range of 100 to 1000 nanometer diameter, then multilaminar large vesicle with size greater than 500 nanometer, then multivesicular vesicles from size 1000 nanometer to several thousands of nanometers. Now principle, it uses a positively charged cationic lipid, or neutral lipid. So what happens is that the genetic material is actually negatively charged, right? So since it is positively charged, these lipids can easily incorporate the genetic material into itself. So that is one advantage. So or it can use neutral lipids. So when we use neutral lipids, the thing is that maybe depending on the pH of the medium or some factors like that, the neutrality of lipids can be altered, and in such way it can be used. So in basis of cationic lipid system is like the negatively charged DNA is that the positively charged hydrophilic head, the head part, the lipid molecule will be, it is having a head, hydrophilic head, and a hydrophobic tail, right? So the hydrophilic head part will be positively charged and it can incorporate the negatively charged. It can condense the negatively charged DNA, and the tail and this head and tail structure, it helps in the formation of a bilayer around the DNA. So this complex, that is DNA plus lipid, it is called the lipoplex, and this lipoplex will protect the genetic material against the nucleases that will be present, that can be present inside the cytosol of the cell, right? So it can protect. So when we look into the methodology, you can see the genetic material and the liposome, the genetic material get incorporated into the liposome, and this is now called lipoplex. Okay. Then this lipoplex get endocytosized and the endosome vesicles are formed. Then the endosome vesicle, what happens? The proton sponge effect, and then the the endosome can go into the proton sponge effect, release DNA, and then the transcription, it can get incorporated into the genetic material. That is one way. And second way is that some endosomes get identified by our lysosome, and lysosomal degradation can also occur. So in the first way, our target is achieved when the endosome vesicle get ruptured by proton sponge effect and the uptake of genetic material into the nucleus occurs, right? So this is the methodology of lipofection. Now advantages: yields high transfection efficiency and works in a wide variety of eukaryotic cells. Low toxicity, it is very simple and stable. Moreover, a number of cell lines normally resistant to transfection by other methods transfect successfully with this method. Now the disadvantage is that the quite low transfection efficiency. As I said, some endosomal vesicle can be identified by our lysosome and degradation can occur. So the transfection efficiency is quite low. Then the dependence on cell division as well as on high rate of endocytosis, not applicable to all cell types, and low efficiency in most of the primary cells are some problems. Now let's move into the next one, that is DEAE Dextran. So DEAE Dextran was the first non-viral vector transfection. As I mentioned in the second slide, see this Dextran, a polymer, it's a polymer, right? So this is actually carrying the genetic material into the cell, right? So here it can be considered as a vector. But since it is a chemical, it is a non-viral vector. So that's why it is called so. So it is the first non-viral vector transfection method used, and it was done by Vaheri and Pagano in 1965. So it is a cationic polymer. So since it is cationic, that means it is having a positive charge, right? So it can readily incorporate the negatively charged DNA. Okay. Then the method is like the DNA is mixed with DEAE Dextran. The DNA Dextran polymer is formed, and when it comes in contact with negatively charged membrane, then the endocytosis or osmotic shock can lead to endocytosis. So osmotic shock is actually given by a DMS or glycerol in the medium. We can use this chemical compounds to induce osmotic shock, or if the entry can occur by endocytosis also. So uptake usually occur by endocytosis. So this is the method, DNA plus Dextran. DNA plus Dextran. So this is getting endocytosized, and then the release and the rupture, then release of genetic material, all this occurs. Now this thing, the genetic material plus the polymer DEAE Dextran, it can be called polyplexes. And now here this is the proton sponge effect. I had already explained it, but this diagram is much clearer, so I will just explain it once more. So here the endosome is there, and into the endosome, since there are positively charged particles inside it, the HCl releases the chloride ion into our endosomes, and this chloride ions provides a negative charge inside it. Right? Now there is a osmolarity difference and water influx occur into the endosome, and this actually creates a tension in the endosomal vesicle and leads to rupture of endosome and release of the endosome, the contents inside the endosome. So this is what the proton sponge effect is. Now the advantages of this method is that it is inexpensive and easy to perform and quick, can be applied to a wide range of cell types, and disadvantages include high concentration of DEAE Dextran can be toxic to cells, transfection efficiency will vary with cell type, can be used only for transient transfection, typically produce less than 10% delivery in primary cells. Now next method is cationic polymer. So here also the thing is same, like the genetic material is negatively charged and we are using a cationic polymer so that it can easily incorporate the negatively charged genetic material. So cationic polymer is different from lipid, that they do not contain any hydrophobic moiety. In lipid we know that there is hydrophilic head and a hydrophobic tail. So this hydrophobicity is not there, and they are completely soluble in water. The cationic polymer examples are polybrine, then PEI, polyethylene imine, and Dendrimers. The ability of cationic polymer to more efficiently condense DNA is exploited. They can condense the DNA very efficiently because it is positively charged and DNA is negatively charged. That is the principle. So here the methodology. we take the PEI and the DNA, it incorporates the DNA and it get internalized into the cell. So here it is mentioned ligand receptor. This is another method of internalization. So what happens is that there will be a surface ligand or receptor. So and this polymer, the carrier, the vector will have a ligand. So this ligand will be complementary to the ligand receptor on the cell membrane, and this complementarity leads to the internalization of the contents, and this is called receptor mediated internalization of ligand labeled polyplexes. So this complex is termed as polyplex, that is DNA plus the cationic polymer. This is polyplex. So this internalization is receptor mediated internalization. And another method is like we can the internalization can occur by endocytosis. So here we have to note that the cationic polymer usually permits the transient transfection. That is, it do not get incorporated into the genetic material. So that is a disadvantage actually. This incorporation into the genetic material may not occur in the cationic transfection method. So the advantages include they are generally stable in serum and are not temperature sensitive, high efficiency reproducible results. Disadvantages include potential cytotoxicity, non-biodegradable, limited to transient transfection studies. So note, compared to the DEAE Dextran, which is also a cationic polymer, right? So this cationic polymer can offer increased complex stability and more reproducible results and higher transfection efficiency. In addition to, while higher molecular weight cationic polymer tend to be non-biodegradable and more cytotoxic than low molecular weight polymers, they show higher transfection efficiency due to the increased polymer to nucleic acid charge ratio. When this polymer to nucleic acid charge ratio is higher, the transfection efficiency also increases. The higher toxicity of larger molecular weight polymer can be reduced by biodegradable cross-linking. And next one is the Dendrimer. These are highly branched globular macromolecules, and it is an example of our cationic polymer. It is capable of interacting with DNA to form small complexes. So let's look into the structure of Dendrimer. So this is a dendron, that is the branch. Okay, so many dendrons join together to form the dendrimer. So dendrimer will incorporate the genetic material and form a complex, dendrimer genetic material complex, and this complex get endocytosized and then the endosome, then endosomal escape occur, then the genetic material will be released into the cytoplasm. So this is how dendrimer work. So we discussed that in the cationic polymer, the incorporation into the nucleus may not occur. So it is transient transfection. Then PEG mediated transfection. So this also, what happens here is that we collect the hyphae or the whatever, then preparation of the protoplast using the enzymes, we prepare the protoplast, then we do the cell counting and we take the required number of protoplast. Then what happens, we treat it with calcium ions and PEG under ice. Then we mix it with the genetic material and then cultivate it, and the transfection will occur when we mix it with the genetic material after treatment with calcium ions and PEG. So this is very simple method. So here what happens is that we use only protoplasts. So first if we are taking fungi or plants, we have to remove the outer layer, and then polyethylene glycol, that is PEG, it is used to stimulate the endocytosis of the genetic material. So here the PEG that we use is usually 8000 Dalton. We are having the final concentration of 15%. Then the calcium chloride is added, and sucrose and glucose is used for maintaining osmotic osmotic buffer agent. Then to reduce the effects of nuclease, the carrier DNA from salmon or herring sperm can be used. So these carrier DNA will interact with the nucleases and prevent the degradation of our target gene of interest inside the cytoplasm. Then the exposure of protoplasts to exogenous DNA in the presence of PEG and other chemicals like calcium can be used. So after that the PEG is removed. Okay, then the intact surviving protoplasts are then cultured. Then the PEG vesicles are less toxic and more resistant to non-specific protein absorption. So that is one of the advantages of using PEG. Now lithium acetate mediated transfection. Lithium acetate LiAC is a reagent used in lithium acetate method of transforming yeast cells. It's usually performed on yeast cell. So this method, lithium acetate will neutralize the cell membrane's charge. The cell membrane will have a charge, right? So lithium cations will neutralize the negative charge on the cell membrane, allowing the plasmid DNA to be uptaken by the yeast cells. So steps include: we prepare the cell, make the cells competent, then add plasmid DNA into the yeast cells, heat shock method to create pores, and then plate the yeast cells. That is, we culture the yeast cells. Then there are mainly two variations, that is rapid transformation protocol used when a small number of transformants are needed, and high efficiency transformation protocol when a large number of transformant cells are to be generated. So this is lithium acetate mediated transfection. So as a conclusion to this video lecture, we can say that there is no single or universal transfection method or strategy that we can use. This transfection strategy depends upon the type of cells that we take, the equipments that we have, the facility, and also the money that we can give for the experiment, right? We need to have proper support from our institution. All these things matters. So while selecting the transfection strategy, we have to analyze that to what degree of efficiency or what degree of cytotoxicity or that can occur in your experiment, right? You have to analyze everything. We have to optimize a lot of factors, and we have to select a suitable transfection method and use it for your experiment. So that's all for the video lecture. Hope you understood things, and if you have doubts, please feel free to ask in the comment box. I will surely reply to it, and do like, share and subscribe. Thank you.